Coming to this PhD research topic, which I am keen
Yeah.
about. So, can you explain in detail and elaborate on your research area, what you have worked on?
Right. So the thing is that first of all, I wanted to do cancer research, right? So I wanted to do something in the field of cancer cells and other things. But the problem with that one is my professor was working, but he was not doing anything in the field of biology. He was doing everything in electrochemistry because
Okay.
he is an electrochemistry guy. So I had to like combine. Previously I had this one of the senior in my lab, Indian
Hmm.
guy. He is a professor in assistant professor in IIT Guwahati now.
Okay.
So he was working in our lab and he had done some research in that field. So when I joined that lab, my professor actually gave me that work and then he said you can continue this research.
Hmm.
So I didn't have a choice of if I had to choose the topic or not. I just had the topic. So I did that one for 3 months or so, the first 3 months, and then I got an idea of how to do things, and then I went on to do my own thing. So my basic research was in the beginning to detect cancer cells using electrochemistry.
Okay.
So how this is done is you need to prepare your electrode. So in
Hmm.
electrochemistry, we use usually three electrode systems. There is a working electrode, there is a counter electrode, there is a reference electrode.
Yes.
So you have to modify your working electrode so that it will either, catch that cancer cell, or it will give some kind of signal by the approaching presence or absence of that cancer cell. So all this is depending on the biochemical reactions. Like for
Hmm.
example, the cancer cell have some kind of receptor on its surface.
Hmm.
So first you need to culture the cell, and then you need to take the cell, and then you need to modify your electrode based on that receptor. Like for example, I used folate receptors.
Hmm.
Folate receptors are usually used for intake of folic acid.
Hmm.
In pregnant women, folic acid is supplemented. So that is because the growing cells need excess amount of folic acid. So cancer cells have the same property.
Okay.
So I used that one, and then I modified my electrode surface with the folic acid, and then I tried to attach that on the cell, and then the folic acid signal, whatever you get the signal, it should go down. It's a negative, like indirect detection. Like if the signal goes down, your cell is attached, something like that. So
Hmm.
based on that, I did my first work was on that one. So I was able to detect the leukemia cells, which actually are really difficult to detect.
Hmm.
So we theorized this strategy for
Hmm.
detecting leukemia cells, blood cancer cells, and then using that, the next step was, this work, like I said before, this was almost 1.5 years to
Okay.
complete. Like first 6 months I didn't understand, and then the thing went for another year. So and then what happened was I wanted to try a different area. Since this went 2 years, I was also working on some other research projects. Like some of my friends were working on, and then there was no biology student in our lab, so I was the
Hmm.
only biology guy. So whatever the biological reactions, I used to do that one. So I worked with some of the postdocs in my lab, and then uh, someone who is working on energy, someone who is working on sensors, chemical sensors, and then gas sensors, something like this. So we did nearly what 5, 6 projects together. There was a
Hmm.
postdoc from Japan at the time. Indian guy. He did his
Hmm.
PhD in Japan, and then he came for postdoc in our lab. And he was very well versed in cancer biology and
Okay.
regenerative medicine because he was working in Japan at the time with the same thing.
Hmm.
So I worked with him on 2 projects for cell lung cancer detection and other things. And I mean, your main project will be going, but you'll be working on some different projects.
Yeah.
So I finished nearly 5 or 6 projects at the time, in combination with this one. And then by that time, like by 2015 or so, I was already fed up with the sensor part. Like cell sensor was already over for me. But the thing is, my professor was interested in continuing that one. So you can't actually say no, because the PhD supervisor is the one who gets funding. So he is the one who's going to tell you. So my interest was shifting towards microfluidics at that time. So because the thing is, you have to have a future plan. I mean, what you are going to do after this one. But the sensor, in case of biochemical sensors, no? So every time you fabricate a sensor and then you try to detect cancer cell or something, the electrode goes to something called as fouling. So your result is not always constant. It keeps on changing. So most of the people find it really difficult to believe the electrochemical sensors, because see, one of the most successful electrochemical sensors is the glucose sensor. Everybody uses, all over the world, everybody uses this one. But after that, there is no development in case of electrochemical sensors. I mean, there is, but not like in other fields. Why? Because the signal that you get is always unreliable. Not unreliable, but it keeps on changing. You know what's happening, you know the reaction, but you can't actually, just get once and then decide, okay, this is fine, this done, finished.
It should be repeatable.
That's not the case.
Reproducible.
Yeah, it should be reproducible, but in order to get a reproducible result, you need a lot of time, like 2 years, 3 years, or something. But in my cases, I had already spent 2 years there, so I had to finish as soon as possible, because after like one more project will take 1 or 2 more years, and then I'm done. So I was interested in microfluidics. So microfluidics, but I had to work with cancer cells because project was there. So my professor suggested that, okay, you can work, but you have to work on this one. Just give me a proposal and then we'll decide. So I wrote a proposal to separate cancer cells from blood using the cell decoration, this one strategy. What it means is that you can put something on the cell which will give you signal, because cell doesn't give any kind of electrochemical signal.
Yeah.
So you can put something on the cell surface which will give you signal, and then you can run through the microfluidic channel, and at the end of the channel you can detect and separate and collect all these things. So I proposed that, but that project got changed a lot, and then I think it took like 8 months to finalize that project, like what we are going to do exactly. And I did I don't know 37 samples or something. We got some patient blood samples from the University Medical Center in Pusan, and then we checked, like some of them were normal patients, like not cancerous patients, some like other diseases, something. And then some were actually cancer patients with metastasis, some were with non-metastatic cancers. So we got around 90% or something, like accuracy with that one. So which was good actually, because it's really difficult to get more than 80, 85%, 90, 95%, something like this. So we got around 90%. So I published that. That work was actually published after my PhD.
Okay.
And again, so like I said, my professor was pushing me towards the cancer cells. So I worked on electrochemical, but cell detection. So I also worked on some other cell detection, like bone cancer and other things. So finally, I also worked on, there is one technique called as CellX. So it's a process of generating aptamers.
Okay.
So we did something with that one using a microfluidic system and one-step generation of CellX. So like I worked on different kinds of projects at this one. Finally, what happened was in my PhD defense, by the end of this one, like I said, you had to have a story. I had to have a story at the time. So I used only 3 papers. The first one, the electrochemical detection of cells, and the second one was the bone cancer detection. And then third one, I used the microfluidics. With the combination of first one and second one, I just used that one, and then finished the thesis. The other, I did not even present any other papers because there is no story with that one. So in PhD, yeah, the one thing in PhD is that every department have some rules. So you have to publish certain amount of papers. For example, in Japan, you don't need to publish papers. You just need to publish one paper. Maybe that's enough. Even in European countries, even one paper is enough. But in my department, we had a rule that you have to publish at least 3 in SCI, more than high, like 5 impact or something, or overall 5 impact or something like that rule. But my first paper actually satisfied all the criteria, so I was okay with that one.
Okay.
Yeah. So finished PhD.
Yeah. So currently you're working as a postdoc at Yonsei University. Yeah. So in which area you're working here?
Actually, when I applied, I wanted to work on microfluidics again because in my previous lab I worked on glass-based microfluidic channels. Now, actually, in my current lab, we are doing PDMS, the polydimethyl siloxane-based microfluidics, which is very good for 3-dimensional cell culture and then analyte detection. So, which is my area actually. So, I actually wanted to work on that one, but my professor has a project for Environmental Ministry of Korea, and that project, not only environmental. Actually, there are like 4 ministries involved, and then it's a very big project. So they have a 5-year project, which we finished now. Just now we finished 2 years of that one. So this one involves the analysis of the entire country's water system using electrochemical sensors, and they are making the automated machines and machine learning, artificial intelligence type of this one. So my job right now is to develop sensors to detect the plasticizing molecules one at a time. So I have to develop sensing systems for one pallet tester or like plasticizer at a time. But later they will combine like 4 or 5 projects together, and then they will try to make an automated system or something like that. But that one, I mean, my contract is up to the end of this year. So, but I already finished my project. So my project is finished. I'm trying to publish that one now. But I think it will go on for the next 2 years. But if I continue, maybe I have to work on the combination of systems. Like now we have 3 systems already finished now. So one of the PhD students in my lab, he actually worked on similar research, and then I worked on that research. So now we have to do combination of the systems, like which is difficult because when you develop a system, isolated system, it will work good on its own, but you are not expecting it to work good with other systems. So, but now when you have to combine, you need to change things like base material, and then like strategies you need to change. So that one, maybe it will go. But right now, since it's a government project, then we can't discuss more. But then,
yeah,
it
will be confidential.
No, I mean, yeah, it is confidential, but the thing is, it's okay because most of the people are doing this. Like whatever I told you right now, it's a general explanation. Most of the people are doing this one. That's okay. But what strategy we are using, that is the main thing.
Yeah, exactly.
Yeah.